Hello students, welcome to EPG Pathshala. In previous two modules, we have discussed about pesticide toxicity and carbon monoxide toxicity. In this module, we shall be discussing about lead toxicity. You all know that lead is a metal and this is widespreadly present in environment because of its excessive use by human beings in different activities. So, the learning objective of this module are to know about sources of lead in the environment, to gain knowledge about lead absorption, distribution and excretion from the human body, to understand about the mechanism of lead toxicity and to know about toxic effect of lead. Lead is a metallic element from group 4A of the periodic table. It is a soft metal with little tensile strength. This metal is very malleable and flexible and a poor conductor of electricity. Before we go into the details of lead toxicity, let me have a look on the properties of lead. The chemical symbol of lead is Pb and this Pb has been derived from the Latin word plumbum. Atomic number of lead is 82, atomic mass of lead is 207.2 and density of lead is 11.3.4 gram per centimeter cubes. It means that 1 liter of lead is equal to 11.342 kilogram. Melting point of lead is 327.5 degrees Celsius, whereas the boiling point of lead is 1740 degree Celsius. Lead exists in two oxidation states, that is Pb2 and Pb4, but it mainly occurs as Pb2 plus. As far as color of lead in metallic form is concerned, this is bluish white. If we come to the isotopes of lead, it has 4 isotopes. Lead has several isotopes. The common isotopes of lead are Pb204, Pb206, Pb207 and Pb208. Only Pb204 is the stable naturally occurring lead isotope. Other 3 isotopes, that is Pb206, Pb207 and Pb208 are end products of one of the 3 series of radioactive decay. The end product of uranium series is Pb206 and end product of thorium series is Pb208 and end product of actinium series is Pb207. As far as ores of lead are concerned, galena is the most common ore, that is lead sulphide ore, then crucite, that is a carbonate ore, that is PbCO3 and anglesite is another ore, that is PbSO4. So, these are some of the common properties of lead. Lead is a extremely toxic metal without any known biological functions. As far as the sources of lead in the environment is concerned, this is having natural as well as anthropogenic reasons. The common natural source of lead in the environment is natural withering of the rocks, and main anthropogenic sources of lead in the environment are mining and smelting of lead ores, metal plating and finishing operations, additives in gasoline, organic lead compound used as anti-knocking agent. You all know in the past we use tetraethyl lead as the anti-knocking agent, but nowadays the use of this compound has been stopped. Instead of this, some new non-metallic anti-knocking agents have been introduced. As far as industrial sources are concerned, paint pigment, that is white lead, lead carbonate hydroxide, inks, these are used for printing newspapers, then glazes, fertilizers, pesticides, then leaded storage batteries, these are used in the vehicles, industries and electricity backup systems. Then ammunition also contains lead, then X-ray shielding, Then crystal glass production, solders, alloys this brass and bronze, they contain lead. Then pesticides and fertilizers, plumbing pipes. This is also used as stabilizer in PVC, and there are many other industrial applications of the lead. During the age of Roman Empire, lead acetate was used to sweeten port wine. In urban areas, lead emissions originate from use of leaded petrol, although nowadays this is banned. Over the last few decades, however, lead emissions in these areas have decreased markedly due to the introduction of unleaded petrol. Organolead compounds such as tetramethyl lead and tetraethyl lead are highly lipophilic with low water solubility. Both tetramethyl lead and tetraethyl lead can penetrate body and cell membranes and may also cross the blood brain barrier in the adults. Exposure to lead. Lead is a multimedia pollutant with various sources and media contributing to its exposure. Although exposure to lead occurs mainly through air and food, the absorption through skin has also been reported by several authors, and exposure to lead through inhalation occurs from the following sources. In urban areas where leaded gasoline is still in use or has only recently been phased out, mean air lead levels are quite high. In the vicinity of lead emitting industries, for example, lead smelters, lead acid battery recycling plants, etc., lead exposure occurs through inhalation as well as ingestion. Additionally, inhalation exposure occurs through cigarette smoking, both in active and passive smokers. Airborne lead gets deposited on soil and water, thus reaching human via the food chain. Exposure to lead through ingestion. Dust, dust including this street dust, home dust and playground dust have been recognized as major sources of lead exposure, especially to young children because of their greater susceptibility to a given dose of toxin and the likelihood to ingest inadvertently significant quantities of dust through pica, that is the mouthing of known food objects, and repetitive hand or finger sucking. Dust around homes painted with leaded paints and around lead emitting industries may contain very high lead. In cities with old plumbing system, use of lead pipes are very common. In such areas, lead is found in tap water or the water that is supplied to our homes. Lead intake also occurs through foods. In diet, cereals, vegetables, fruits, bakery products and beverages, the presence of lead has been reported, and it has been found that lead is usually present in the food items. Food and vegetable, these fruits and vegetables, they contain lead as a result of deposition from air as well as uptake from the roots. Lead acetate was previously used as sweetener in the wine, that was also a source of lead in the body. Wines may contain considerable lead concentrations, as lead arsenate was used as a fungicide in vineyards, and lead contamination from containers may also occur. Another significant source of lead is lead glazed or lead painted pottery. Particularly storage of acidic foods such as fruit juices, pickles in lead glazed ceramic may lead to large lead intake. Exposure to lead via skin. Although lead mainly enters the body via oral ingestion or inhalation, the absorption through skin has also been reported by several authors. Exposure to lead may occur from use of cosmetics also. Some of the common cosmetics that contain lead is leaded eye powder, that is surma or kohal. Skin penetration of lead may occur in occupationally lead exposed workers. Now, occupational environment and lead. Occupational exposure to inorganic lead occurs in mines, smelters, welding of lead painted metal and in battery plants. High levels of air emission may pollute areas in the vicinity of lead mines and smelters. Occupational exposure to lead may be categorized in high risk and moderate or low risk categories. This table shows the occupational exposure to lead. Exposure may be high risk exposure and moderate or low risk. In case of high risk, exposure may be from primary and secondary lead smelters, production of lead paints, spray painting using lead paint, flame welding and cutting of lead painted metal, ship breaking, brass foundry work including bronze, ammunition work, storage battery manufacturing, addition of lead stabilizer or lead to PVC, blasting or scrapping of lead painted and indoor shootings. So, these are some of the occupational exposure that cause high risk. As far as moderate and low risk occupational exposure are concerned, they include lead mining, cable industry, type casting in printing shops, lead soldering, that is tin soldering, porcelain manufacturing, crystal glass manufacturing, earthenware manufacturing, glass painting, plumbing, stereotype composing, lead casting, car repairing, electric welding in lead painted metals, sulfuric acid production, application of lead arsenate pesticides and wire drying. So, these are some of the occupational exposure to the lead. Let me have a look on lead absorption, distribution, elimination and excretion. Lead is absorbed through respiratory tract, gastrointestinal tract and skin. Adults may take up only 10 to 15% of lead in food, whereas children may absorb up to 50% of lead via the gastrointestinal tract. So, we can say that absorption of lead in children from gastrointestinal tract route is more than the adults. Gastrointestinal absorption of lead is affected by nutritional status. Milk and vitamin D increase the gastrointestinal absorption of lead. Lead distribution in the body. Lead is one of the systemic poisons. Once absorbed and comes into the circulation, it is distributed throughout the body, causing serious health effects. Most of the blood lead, that is written as PB, is present in the erythrocytes, that is red cells, leaving only a fraction of less than of 1% in plasma. Lead's high affinity for the protein D-aminolevulinic acid dehydrates, that is ALDA, equivalent to porphobilinogen synthase, that is PBGS. ALAD is an enzyme present in all cells, including the erythrocytes. It is the second enzyme in the heme pathway, promoting the asymmetrical addition of two molecules of D-aminolevulinic acid to form the monopyrrole porphobilinogen. Absorbed lead from the blood plasma is distributed to other organs. Among the soft tissues, the liver and the kidney attain the highest concentrations of lead. In adults, inorganic lead does not penetrate the blood brain barrier, whereas this barrier is less developed in the children. So, in case of children, this blood brain barrier can be crossed by the lead. The degree of passage of lead through blood brain barrier into the nervous system is higher in infants and children than in adults. So, children are especially susceptible to lead exposure and subsequent brain damage due to high gastrointestinal uptake and the permeable blood-brain barrier. A large fraction of the absorbed lead, approximately 90%, is incorporated into the calcified tissues such as skeletons, bones and teeth. Bones act as a reservoir for lead, and a two-way process of active influx and efflux of lead to and from the bone and blood stream has been recognized. Bones are no longer considered as sink of lead in the human body. And let me have a look on the lead elimination or excretion from the body. Lead is excreted from the body mainly through the urine and feces. Lead is also excreted through bile and pancreatic juices into the feces. To some extent, lead is also excreted in saliva and sweat, but is of little importance. There are several biomarkers for lead. One is heme synthesis. Lead inhibits several enzymes in the heme synthesis, primarily D-aminolevulinic acid dehydrogenase and coproporphyrinogen oxidase. The effect of lead on heme synthesis causes accumulation or increase of D-aminolevulinic acid and coproporphyrins in serum and plasma and are also excreted into urine. This ALA is a neurotoxin. Then nucleotide metabolism can be another indicator. P5N is present in the cytosol of erythrocytes. It catalyzes as a step in degradation of ribosomal RNA, hydrolytic dephosphorylation of pyrimidine 5 monophosphatase. Lead inhibits P59, that is pyrimidine 5 nucleotidase enzyme activity, which leads to accumulation of pyrimidine nucleotides in erythrocytes, that is red cells, shortening the lifespan of cell. Lead also interferes with calcium dependent processes. This includes such as related to neuronal signaling and intracellular signal transduction. Toxic effects of lead poisoning on human health. The symptoms of acute lead poisoning are headache, irritability, abdomen pain and various symptoms related to nervous system. The characteristics of lead encephalopathy are sleeplessness, restlessness, and in severe cases the affected person may suffer from acute psychosis, confusion and reduced consciousness. Children may be affected by behavioral disturbances, learning and concentration difficulties. Long-term exposure to lead may lead to memory deterioration, prolonged reaction time and reduced ability to understand and anemia, as lead poisoning causes disturbances of hemoglobin synthesis. This table shows the toxic effect of lead poisoning on human health. Let me discuss one by one. Let first we take nervous system. Lead affect central nervous system, peripheral nervous system, and it has very complex effects on the nervous system. As far as blood is concerned, lead affects heme metabolism. Heavy lead exposure may cause anemia, and this also affects nucleotide metabolism. Lead is having its effect on kidneys also. Severe exposure to kidneys may cause renal tubular dysfunction and resulting in hyperuricemia, hypertension and gout, and chronic renal failure may also be there. Lead may also affect cardiovascular system. may affect endocrine system, immune system, and also may have mutagenicity, means may cause chromosomal aberrations, and it is well known that cancer may cause kidney and lung, lead may cause kidney and lung cancer. This may also affect the reproduction in male as well as females. This may affect sperm quality and fertility in males, and in case of females, lead can cross placental barrier and may reach the fetus, resulting in stillbirths, abortions and miscarriages. As far as effects of lead on gastrointestinal tract are concerned, they are also evident. This may cause nausea, abdominal cramps and constipation. In nutshell, we can say that lead affect a number of functions and body organs. Now, the question arises, what is the mechanism of lead toxicity? You know, and you have learnt that lead toxicity is multifaceted. Various mechanisms of lead toxicity are there. Let me discuss these mechanisms. These mechanisms includes interruption in activity of enzymes, then ionic mechanism is there by which lead toxicity is caused, then deactivation of antioxidants, sulphadryl pools, then free radical damage and oxidative stress. Interruption in activity of enzymes. Two specific enzymes, these are D-aminolevulinic acid dehydrogenase and glutathione reductase are inhibited by lead. ALAD is primarily involved in heme synthesis. So, inhibition of ALAD may by lead increases levels of the substrate, that is D-aminolevulinic acid, which is known to stimulate the formation of ROS. Glutathione reductase is an enzyme that is responsible for recycling of oxidized glutathione, and lead has been shown to interfere with this cycle, resulting in depressed GSH levels. Under normal conditions, GSS, that is reduced glutathione, and GSSG, oxidized glutathione, are present in the ratio of 90:10. Under the condition of oxidative stress, the GSSG concentration exceeds the concentration of GSH. Then if we come to the ionic mechanism, the divalent lead ion is similar in many ways to calcium ions. The ionic mechanism of lead toxicity occurs mainly due to the ability of lead metal ions to mimic with bivalent cations like calcium, magnesium and iron in several biological systems, ultimately disturbing the biological metabolism of the cell. Lead also interfere with calcium dependent processes such as related to neuronal signaling and intracellular signal transduction. Significant changes in various biological processes such as cell adhesion, protein folding, intra and intercellular signaling, maturation, apoptosis, enzyme regulation, ionic transportation and release of neurotransmitters are caused by the ionic mechanism of lead toxicity. Due to chemical similarity between calcium and lead, lead can substitute calcium, which may account for the fact that 90% or more of the total body burden of lead is found in skeleton. Then another mechanism is deactivation of antioxidants, sulphadryl pools. Lead is an electropositive metal and has high affinity for the sulphadryl, that is SH group. Lead react with sulphadryl group of the enzyme to form mercaptide, leading to enzyme inactivation. Here this reaction is shown. Free radical damage and oxidative stress is another mechanism. Transition metal homeostasis is maintained through tightly regulated mechanism of uptake, storage and excretion. Exposure to lead can break down the metal ion homeostasis, which can lead to enhanced formation of reactive oxygen species ROS and reactive nitrogen species RNS, involved in causing oxidative damage to biological macromolecules such as proteins, lipids and DNA. Imbalance between the generation of free radicals, that is ROS and RNS, and their detoxification by antioxidant defense system, that is non-enzyme antioxidants such as glutathione, ascorbic acid, flavonoids, alpha tocopherol, carotenoids and enzyme antioxidants, which include glutathione peroxidase, superoxide dismutases and catalases, is termed oxidative stress. Free radical induced damage by lead is accomplished by two mechanisms. These two independent although related mechanisms are direct formation of ROS, including singlet oxygen, hydrogen peroxides and hydroperoxides. Simultaneous depletion of the cellular antioxidant reserves. Reactive oxygen species ROS are generated by various endogenous and exogenous sources. The main endogenous source of ROS is mitochondria, along with microsomes and peroxisomes. These generate specially H2O2. Atmospheric pollutants, tobacco, smoke, then irradiation by gamma rays, X-rays, UV, etc. Then heavy metals and chemicals are some of the exogenous sources of ROS generation. Reactive oxygen species, that is ROS, can be classified into two different groups. Free radicals, also known as pro-oxidants, possesses unpaired electrons in their outer orbitals. Superoxide anion radical, hydroxyl radicals, hydroperoxyl radical, alkoxy radical and peroxy radical. Then non-radical oxygen species. Very reactive, also possesses unpaired electrons and are capable of forming ROS radicals. Organic hydroperoxide, hydrogen peroxide, ozone and trioxidine are some of the examples. Under normal conditions, ROS are considered as redox regulators and are fundamental keeper of cellular homeostasis, playing essential functions as signal molecules in several signaling pathways involved in cell differentiation, organogenesis, stress response and wound healing. Under oxidative stress, the free radical induced toxicity at cellular level is expressed as lipid and protein peroxidation and damaged nucleic acids. This figure shows heavy metal induced oxidative stress. Lead may also affect the plants, because plants strongly inhibit the key enzyme of chlorophyll biosynthesis, D-amino livulinate dehydrogenase. In most cases, inhibition of enzyme activities is exerted through interaction of lead with enzyme SH groups. However, activities of some enzymes may be increased by lead, for example, chlorophyllase activity, which leads to enhanced degradation of chlorophyll. Lead reduces the uptake of nutrients, namely cations, that may be potassium, calcium, magnesium, manganese, zinc, copper, iron, etc., as well as of anions, for example, nitrate. Lead also promotes formation of reactive oxygen species in plants, leading to oxidative stress. Lead toxicity symptoms in plants are chlorosis, stunted growth of the plant, blackening of root system of the plant, inhibited photosynthesis. Lead also affects membrane structure and permeability of the membrane. Lead may upset mineral nutrition and water balance and may also decrease in hormonal status of the plant. Lead also inhibit germination of seeds and hence germination percent and retards seedling growth, root shoot length and dry mass of the plant. So, in nutshell we can say that lead has severe effects on the plant growth, plant germination as well as plant yield. So, dear student, in this module we have learnt about various properties of the lead, what are the different sources of emission of lead, and we have learnt about the effects of lead on human beings, and we have also learnt about its accumulation in the body and its excretion and elimination from the body, and we have also learnt about the effects of lead on the plants in this module. Thank you.